Hello, everyone. Welcome to SECOORA's webinar series, Coastal Observing in Your Community. Thank you so much for sharing your lunch hour with us. We are hosting this webinar series to discuss, highlight, and raise awareness about coastal ocean observing activities in the Southeast U.S. My name is Abby Wakely, SECOORA's Communications Director, and I will be facilitating and moderating this webinar. All participants will be in listen-only mode. You are encouraged to type questions for the presenter or technical questions for me in the chat box. I will respond to them directly or post the questions to our presenter after his presentation. We are recording this webinar today and will be posting the recording along with the PDF of the presentation on the SECOORA website. We are very excited to welcome Dr. Frank Muller-Karger from the University of South Florida College of Marine Science. He will be presenting Observations to Understand Life in the Ocean, linking IOOS regional efforts with the Marine Biodiversity Observation Network, known as MBON. Dr. Frank Muller-Karger is a biological oceanographer who conducts research on marine primary production and the diversity of life in the sea. He combines traditional oceanographic methods and satellite remote sensing to study patterns of phytoplankton distribution and how these patterns change over time. He studies the oceanography of the Gulf of Mexico, the Caribbean Sea, the Atlantic Ocean, and also has some work of global scope. He is involved in a number of international working groups and holds a Bachelor of Science, Master of Science, and PhD in Marine Science, in addition to a master's degree in management. Dr. Muller-Karger has authored or co-authored over 160 scientific publications. Dr. Muller-Karger, thank you so much for joining us. I'm going to go ahead and promote you to presenter so you can share your screen.
Well, thank you, Abby, and the people from SECOORA for inviting me. This is a presentation that I'm doing jointly with Enrique Montes, who's also on the call, and Francisco Chavez from MBON. And the background for this is a project that is called the Marine Biodiversity Observation Network. There's 3 of these in the U.S., and there's a 4th one really that is Marine Geo from the Smithsonian. And together we are these pilot projects that we call MBON in the U.S. This one in particular grew out of an effort that we're doing with the National Marine Sanctuaries Office, and in particular the Florida Keys, Flower Garden Banks, and Monterey Bay. And I'm going to focus on the southeast region of the U.S. because this is really one area that we would like to see how we can get biological observations engaged more fully into the observing system. So what I'm going to do is I'm going to talk a little bit about why we do this, some of the new technologies that allow automation of biological observations, and then a strategy for engaging IOOS and bringing those observations out to stakeholders. So as you in SECOORA go through thinking on how and why we're going to do this, is really you need to think about who requires which biological data, what technologies and what approaches can we use, what's available that is sort of low-hanging fruit to make this data available, and then why we're doing this. And really the reason is that we need to measure change of biological resources in a way that benefits people, and that we can understand why the ocean is changing, and as the ocean changes, of course, life in the ocean is changing. And this biological and biodiversity data is really critical to understand these resources that we use continuously and actively, and to come up with good ideas and good ways to continue to develop the coast and the ocean in a way that also conserves these resources. So this is a very fundamental, almost very simplistic statement, but this really what drives a lot of things that we do in collecting ocean observations anyway. If you think about why we collect salinity, temperature, current data, wind data, in many cases, this is to understand the impact of environmental change on biological resources, be it fish or marine mammals, or how they move around, or primary producers and how their patterns change in time and in space, and ultimately humans, and we're just as part of an ecosystem as anybody else. So all of these measurements are intended to, in some way or other, support the understanding of biology. So as we go through, and as IOOS, and in this case particularly SECOORA, thinks about why and how to do this, there are several tools. The community has come a long way in trying to understand how to bring biology into an observing system. So one way is to use this framework that is called the Framework for Ocean Observing that came up with essential ocean variables. And there's a whole list of biological EOVs that are these are some of the things that oceanographers have measured for a long time, but they are really now put in a way for a specific purpose to serve specific stakeholders, and in linking them with other essential ocean variables, specifically physical and biogeochemical variables. And so the idea is to measure some of these things together and add biology to existing observing systems. So the problem is that traditionally biological observations are slow to get, they are very time consuming, and so you end up with limited coverage both in space and in time, and so you end up with very gappy fields and maps and time series. And yet we still don't have a good way to generate quote-unquote products that people can consume. In many ways, there's now this idea that we can develop indicators, be it for the Sustainable Development Goals, or for HE targets, or for just simply trying to understand how conditions in a sanctuary are changing. So how can you distill many variables into one or a few variables that we can call indicators is a tricky thing. And so there's a lot of discussion that has to happen about that. Then the data latencies, how quickly people can move biological data from collection from the field into a product that is consumed publicly is a big issue. Much of the biological data that we have collected historically is collected by hand, by boats. It takes a lot of lab time, a lot of quality control, and it takes sometimes years to have these data get out to the public. And so it's very hard to measure change if the data is not available for 10 years or 15 years. And so that's a really serious issue that we have, not only in the US, but internationally. Then we're coming up with how do you manage this data? How do you curate it? How do you archive it? How do you serve it to people? There's a whole issue of sharing data that we have to come to grips with. The physical oceanography community has kind of dealt with this already for the past 20, 30 years. They have realized that if they share data, they can understand weather better, they can bring it into models better, they can really try to understand how things are changing in a real-time basis. And for biology, we have not, as a community, we have not really come to that agreement and that way of thinking. So how do we do that? Maybe for a few biological variables that can lead to indices that we can share rapidly and openly and try to understand how life in the ocean is changing. And then ultimately, there's a whole issue of costs, and making some biological measurements can be very cheap, but some, if you try to make them at the bottom of the ocean, or if you try to make them farther offshore, can be very expensive. And still today, some of the automated devices that we have for measuring biological variables are very expensive. So these are things that we have to hopefully come to grips with, and over the next decade have a revolution in the way that we do things. This is an example of the Global Ocean Observing System framework for ocean observing, and I adapted it here for the IOOS framework, in which we have on the left-hand side, IOOS is developing requirements, and that's what each one of the regional associations does, working with stakeholders, as you identify what a group, a person, an institution may need as a requirement to satisfy a need, and then you develop the product list or the type of variables that you have to collect. In this case, what we're talking about are biological variables that either MBON or the Animal Telemetry Network can collect together. These are both our networks that try to bring the community that deals with ecology or marine biodiversity observing together, or that brings animal telemetry ideas together or data and pushes it out to the community. So the concoction of indices and such would happen in this box, and this would happen at each one of the regional associations or at a broader scale with MBON nationally or internationally. And then you work through a process to entrain the data into the IOOS data management and communication system. And what we have worked with IOOS is to come to an agreement to use the data schema that are used by the Ocean Biogeographic information system, or OBIS, that is called Darwin Core. And I'm not going to talk a lot about Darwin Core, but Darwin Core is simply a structure of data where you have a table with specific rows and columns that are labeled in a specific way, and it's not very complicated, but it is at least a standard, and it's very well curated, and you can push it out to the public, both in the U.S. through U.S. OBIS, which lives in the U.S. Geological Survey, or internationally with OBIS that lives under the Intergovernmental Oceanographic Commission. So there's a big platform to serve data internationally. And then you have a loop to feedback if the products are good or bad, or you try to see how users use these data sets. There's a lot of technology that has come up in the last 20, 30, 40 years that is available to make some biological measurements in an automated way. There's a lot of traditional methods that we should not abandon by any means, but there are ways now to collect data either as a flow-through system on boats, or anchored on a mooring, or even on gliders and other automated systems like Argo floats. And so some are target very small organisms like microbes and phytoplankton, and so that's, for example, an imaging flow cytobot that is an optics device, is a microscope that you can go on a mooring, or you can put on a ship as a flow-through system, and it gives you the diversity of phytoplankton cells. And you can do similar things with zooplankton, and I'll show a couple of examples. We can do that with fish, with the benthos. We can do that now also with animal tracking. Animal tracking is interesting because it not only allows you to see where an animal is going, there are sensors that you can attach to animals and collect real live environmental data as the animal is going through its movements and as it goes from one place to another. And that can then be retrieved in near real time. Acoustics is a very old technology that we can use to collect data about zooplankton concentration. We have fish active and passive acoustic sensors now that can help track fish, but also detect them and maybe even measure abundance. We have tags for top predators, and we have also ways to scan the bottom using sound. We have a whole family of of technologies that deal with genomics, and I'll mention environmental DNA later on, and so that you can sample from microbes to marine mammals using these new methods. And these are very rapidly evolving methods, but they cover a broad number of topics, including abundance and identifying organisms, all the way to what these organisms are doing, basically expressions of traits. And we have also platforms like not only ships, but we have automated underwater vehicles, floats, of course, moorings, and satellites that are extremely powerful, and we can use to look at many of these different trophic levels. And ultimately, we have numerical models or simulations that allow us to understand phenomena, or to even make short-term or long-term forecasts of how the ocean is behaving. And yet we're way behind in integrating biology into these models because biological systems are so complicated. So the higher up we go in the food chain, the harder it is to really model organisms and where they are, or how much they produce, or what the impact may be of fishing. But these are things that are actually happening. And integrating all of this is, of course, the job of IOOS. So I'm going to show you some examples of the technologies that are now available for automated biological observations, and by no means this is supposed to be exhaustive. It's just really kind of a very simplistic sampling of some of the devices that are available. One of the ones that is very exciting is this, the flow cytobot. It's an imaging microscope that also is a flow cytometer. So it basically takes pictures of these single-celled organisms, and it also measures some things like fluorescence. And with these, you can identify the species, sometimes not quite the species, but you can follow them in time. And Heidi Sosek at Woods Hole Oceanographic Institution has had one of these things anchored for many years in Martha's Vineyard. So you can see how things change, not only seasonally, but because other parameters are changing. So if you look at the lower right-hand side, lower right-hand corner of the graph, you'll see that there's some colored plots with some diagonal lines. And what the first plot is temperature on the lower right-hand corner, left-hand side. You can see that the downward slope of the line means that the seasons are starting earlier. Basically, you reach warming earlier in the year, and then starting about 2013, it started again warming later in the year. And organisms like these phytoplankton that they were measuring with these devices followed that the same way. So they followed the spring bloom, the spring stratification and warming of the waters, and so the spring bloom started happening earlier and earlier until 2013, and then it started going back again. So you can track changes in the season or the phenology by looking at these detailed pictures of organisms. There's something similar like that, but for zooplankton. So you can span a broad range in organisms from very small size, with cytobot, to this thing called the underwater vision profiler that can give you measures of zooplankton. And it's the same thing. It's an imaging device. It allows you to classify organisms automatically, and then you can send it out to the cloud for sharing with other people. So this can be attached to the CTD, for example, or it can be dangled from a cable on its own. Now, there's a traditional technology. This is the continuous plankton recorder that has been deployed for almost 100 years in some places, and it's towed behind a ship. It collects zooplankton on a little film strip, but it also collects phytoplankton. And so there's a big effort trying to understand how zooplankton and phytoplankton have changed over time over these shipping lanes over many years. And the problem with this is that it really is tied to shipping lanes, and if you look at a map of where these data have been collected, it's very much focused on big shipping lanes in the northern hemisphere, in the North Atlantic, and in the North Pacific. So there are big gaps in the coverage of where we have data of this nature. Then we have acoustics. So you have all sorts of acoustics that you can collect from sensors that you can attach to a buoy, or you can attach on a boat, from the simple fish finder to something more sophisticated like acoustic Doppler current profilers, or ADCPs, where you can not only measure currents, but you can also try to get some measure of zooplankton and maybe even fish. You can look at passively in sound and look at whales, or you can look at all these organisms in an active way. So this is an area that can expand enormously. Even just listening passively to sound, or what we would call noise, and the soundscape in an area may be important in how you characterize the biology or the diversity of what's there. This is an image from the Animal Telemetry Network. So they're developing this portal as part of the ATN at IOOS, where the idea is to serve data about animal movement and also environmental data collected by organisms. So the idea here is to understand migration corridors, breeding behavior, feeding behavior, and if you aggregate everything in different organisms and how their tracks overlay in space and in time, we may be able to understand how biodiversity patterns change, or understand where and when we have biodiversity hotspots. Environmental DNA is very exciting. It's a technology whereby you simply take some water, filter it with some standard methods, and then you can pass it through molecular techniques. And if you use different primers or these pieces of DNA of genes that are called like 16S or 18S or CO1 or 12S, they can target different groups of organisms. So if you use different pieces of DNA, you can basically map what's in a liter of water or a drop of water that or what organisms have been in contact with this water, from microbes to fish and marine mammals. And so those tests of how try to understand exactly what you can get from this type of technology is now going on. You can actually also take eDNA methods where you target specific species and you search in whatever water body you have for something like an invasive species and identify it, and the method would be extremely sensitive for a specific species. eDNA, we cannot see any other way, and this is proven now. There's plenty of papers that show that this is true. And yet it's an expensive method, it's complicated, the technology changes all the time, so it's hard to deploy it operationally at this point. And yet one thing that people can do is you can collect samples and preserve them for future analysis. So that's an important concept that we need to think about, how or whether we do this as part of the data collection that SECOORA or any of the regional associations does, is can we preserve samples for future analysis and develop time series that way. There's also ways to put some of these devices on automated platforms like the long-range AUVs with the environmental sample processor, which can sample on almost anything, including some technologies to put eDNA samplers on some of these AUVs. And now we have a big push to have some of these sensors on floats that are completely automated. They go up and down in the ocean typically to 1,000 meters and come up to the surface and transmit the data. And some of these are now being designed to go deeper. And this program called Biogeochemical Argo can measure some of these variables that are very relevant to biology, including oxygen, nitrates, and some nutrients. But there's also an effort to put zooplankton imaging and eventually some phytoplankton imaging devices on these BGC Argo floats. And so that's a very exciting development, and you can see that there's quite a few of these already out in the ocean. We also use satellites, and there's a push to have better in satellites in a way to have better time resolution, so more frequent observations, better spatial resolution, and better spectral resolution to try to understand the diversity in colors in the ocean and how that relates to biology. And so we've, as part of MBON with Maria Cavanaugh at Oregon State University, we are going through a process of doing classifications where we take different types of physical observations like temperature, light, winds, some of the physical variables that you can measure by satellite very quickly and over very large areas, and combine it with some properties that are more related to biology, like color, different colors, and including what we call fluorescence, which is a measure of phytoplankton, and try to understand how the ocean looks at different times in what we call biogeographic areas. And so we call them seascapes. And the idea here is that you can actually track seascapes in time. So it's not a static biogeographic scheme. It is actually something that changes from month to month and over the years. And so we're trying to validate these seascapes both off the West Coast and off the East Coast. And the Southeast, we're working with IOOS, with Chris Kelly specifically, where we go out in the field every couple months and sample the area with their ship. And we also are working with the Florida Keys National Marine Sanctuary from smaller boats and collect data and very near shore to see how these seascapes can affect the environment in Florida Bay, or what does it mean when we look at these different biogeographic areas in terms of actual samples of biological actual biological samples. Like in this case, we're comparing to phytoplankton concentration and taxa, and what we see is that yes, definitely there's a separation in that different combinations or different communities of phytoplankton live in different biogeographic areas. So that's an area where we're pushing it. We're trying to understand how how these seascapes also map to eDNA patterns, to zooplankton patterns, and so on. So it's very exciting and interesting to do it in different places. We're also using satellite data to work on what we call an early warning system, where we can automatically detect if there is patterns moving from one place to another. For example, the Florida Bank is interested in in detecting coastal and shelf waters moving over the flower garden banks, and we're working with them on a tool that automatically tracks turbidity in the water and see if it's moving toward the sanctuary, and then sends an alert like an email saying, yeah, there's a wave of turbid water moving this way. And we have a dashboard, a web page with dashboards for a river discharge and coastal turbidity, temperature, and then this idea that you can detect an anomaly moving in space over time. So one of the most exciting tools that we have right now, and many of you have heard about this, is the portals where you integrate all of these things. So the portal for SECOORA is managed by Axiom Data Science, and Axiom takes data from all of these different entities that are on the left of the slide, where you can access data from NOAA fisheries or from local state DEP or Fish and Wildlife agencies, and then move it through a mapping interface that can be either large scale, like global or regional, and then also we're developing some ideas on how to facilitate the use of these data in what we call infographics. And so these are cartoons that very simply show different species or different environments or a thermometer, and you can click on it and you can get a time series and detect change. So you don't have to go through a very complicated analysis or reanalysis or reconstruction trying to search through all these data, which is something we found managers don't have the time to do, and that they want a pre-constructed product that they can go back to over and over, or that automatically sends an alert if there's a change beyond a certain threshold. And so these products are intended to support reports like the National Marine Sanctuary Condition Report or Sustainable Development Goal targets or Aichi targets. So from local to global is sometimes you have the same questions and you can scale them up. So this portal is now live, and we are looking for working with SECOORA to develop the infographics for specific habitats. For example, coral reefs is something that we already have for the Florida Keys, but there may be other habitats that we may want to capture in a simple infographic to track specific variables. And so defining what are the variables that we want to track, whether the data is available, is the data available in a way that is curated so that you can understand where it came from, and whether it's good or bad, or you don't really have a way to track its provenance, is very important. So this is the kind of thing that the MBON portal serves at the moment. For the Keys, we have the reef fish visual sensors, or the RVC data, and we have satellite data. We can look at the time history and anomalies of not only the environmental data like chlorophyll or temperature, but we can lay that over the changes in fish abundance in the reef and biodiversity as well. And so you can take, once you construct one of these views, you can save the URL and you can mail it to somebody, and they will click on that URL, and the same analysis that you see will be reconstructed. Then they can still manipulate it and change it further. So that's one of the things that we're going to, is that you can build a story and then mail it to somebody, and they can see the story digitally in a web page and then still manipulate it and enhance it. And the idea is that these products would migrate and then, quote unquote, automatically construct a report. That's the vision for example, the integrated ecosystem assessment that many regions are going through, that is led by NOAA, and also the condition reports for the sanctuary. This is another view. In the left-hand side, we have the infographics. This is for California, but we're going through the process of doing that for other environments, where you can click on an item, and then you get the time series, and you can set thresholds where you would like to have the alarm set. You have views like in the middle, where you have several time series that you can stack. And we also have a product that we developed for the whole world to try to see how, for example, if you're interested as a country in your exclusive economic zone, can we develop some of these indices in terms of temperature, chlorophyll, and link to the international IOOS data set that has historical data on whether an organism or a set of organisms may be present or not, or whether you have endangered or threatened species, and then we have an indicator, for example, the number of threatened species relative to the total amount of species within your EEZ. And the idea is to go to scale it where a user can draw their own boundaries for an area of interest, or to have something like a shapefile that defines the boundaries of a marine protected area, so you can track over time what the balance of different species may be. So these are things that are actually happening right now, and it would be very good to have the engagement of the SECOORA community as we go through. So SECOORA is well prepared to do this. They're working with partners to identify candidate data sets, but I think that we need to have an active effort to really look at what are priority data sets and what is available, whether there's good time series or not. And Axiom is already trained, and they're willing and able, completely engaged with SECOORA to transform these data into the Darwin core format and then serve it out to ERDA into the web, and then it can be harvested by OBIS internationally. So the data management that we have come up with as part of MBON is more or less as follows, and in which each regional association serves as a regional coordinator to support and work with the stakeholders or data collectors of biological data, be it academic, state, or federal, and then go through and transform the data into this Darwin core format. We understand that not everybody is able or has the time to do this, but IOOS is working with Axiom and with the regional associations to entrain, and they have provided some funding to get priority data enrolled into Darwin core through this process. So working with your regional association is the way to go. They will hold your hand. They will do this for you. And ideally, we can set a process where new data, whenever it appears as part of a time series, gets enrolled automatically and then gets served through ERDA through the web. We are interested in I would really like to see it move forward, is that the ATN and the MBON and each one of the regional association web portals are linked better than they are right now. And so designing and exercising those linkages is something that has to be done with a lot of thinking. And for example, the ATN portal right now is moving towards serving animal telemetry network data, but it doesn't have much environmental data. So how in a lot of the MBON, a lot of the environmental data, like maps of sea surface temperature or the other data that you serve through your portal at SECOORA, is not yet linked with ATN. So how do you do that? That's something that I know IOOS is thinking about, but if you can provide feedback on what you would like to see, that would be very, very important, very useful. Then linking all of this and moving the data through a process that it can be served internationally through OBIS would be extremely useful as we consider how we look at change in life in the ocean on an international level or through large regions. And right now, that's not easy, and not a lot of data is moving into OBIS, at least not all the data that we would like to see. There's a lot of old data that many of us may have that has not been touched for a long time. So I wonder can there be a data archaeology effort for priority biology data that we can move through the same process to rescue it, digitize it, and put it out and serve it through a Darwin core. So it would also be nice to see better integration for some of these biological products and environmental products between different regional associations, and the idea of interoperability has been part of IOOS since day one, and yet you still have a disconnect between how you view data. So if I wanted to see a specific data set, if I zoom out to look at the whole East Coast, in many cases I cannot see that through these portals. And so how do we move there if we want to track how organisms move because their range is changing over time, or because you have an organism that is migrating like a whale? How can you integrate different data sets from the different portals into understanding how these organisms are moving? And then designing and using infographics as user-defined data views will require involvement of the people that know the data and that need the data, so that we have served the right product at the right time. So my recommendations for IOOS and SECOORA is that you continue to define specific user needs for biological data, as opposed to just including something or a nice graphic because it's beautiful or because it looks nice. This really has to serve an operational purpose. It has to be very, very directed. Integrating biological observations into IOOS, the time is right to do that. We have talked about it for a long time, at least 20 years that I'm thinking of we should do this, and it's still not there, but I think the technology, including the data management technology, is now there. There are standards that we can use, and so we can do this now. And there's a willingness at NOAA and the interest to do this. There's all kinds of platforms, not only to collect data, but also to deploy these instruments that we can use. It's important to look for customers within NOAA as you think of how to promote observations. All the different lines at NOAA need biological data. So how do you serve those needs is going to be something that we really need to think through. I think that there's a big opportunity for IOOS in NOAA in general to get better or more involved in defining the specifications for new satellite sensors that NASA may develop, either for research or eventually for operations. For example, this technology that we call H4, or high spatial, high spectral, high temporal resolution, and also high quality data. And that's what we need to look at biodiversity in the surface ocean. And that still doesn't exist. And of course, developing integrated biological and physical coupled models that are designed to satisfy specific user need. So these are complicated tasks, but I think that they're very important for IOOS to move forward. And well, that's what I have for now, and I'd be happy if we can have some questions or some discussion.
Great. Thanks so much, Frank. What a wonderful presentation. Now we're going to go ahead and open it up for questions. You guys can type your questions in the chat box or the Q&A box, and I will be asking them to Frank. All right, I'm not getting any in, but I do have a question, Frank. The seascape validations in South Florida, will there be a paper published on this research?
Yeah, I can let Enrique talk about that, but there will be more than one.
Great.
Enrique, do you want to talk about that?
Hi, Abby. Yes, we're actually finalizing this manuscript, and basically what we're doing is that we're collecting seawater samples around South Florida waters, and each of these samples corresponds to a very specific seascape class. So Frank, I don't know if you can go back to the slide that is showing the type of phytoplankton that is per seascape class. That's going to be helpful.
That one?
It's right there. So you can see in this slide that we collected surface samples during 3 cruises, one in March, May, and September 2016, and we measured the phytoplankton pigments using HPLC, the HPLC technique, which gives you a sense of the community structure. You can basically understand how different groups of phytoplankton are contributing to total chlorophyll concentration, and those could be chlorophytes, cryptophytes, diatoms, haptophytes, and so on. And then, so we've been able to get a sense of the relative abundance of these different pigments on these different seascape classes. And we are getting, for example, that in seascape classes that are characteristic of oligotrophic conditions, like seascape number 10 in the pack plot that you can see in the bottom, are mostly dominated by cyanobacteria, and that in very coastal seascapes, that are the ones that are, red and yellow colors on these figures, a big chunk of the community is dominated by diatoms and larger phytoplankton classes. So this,
Wonderful.
yeah, this should be, we're finishing up this manuscript soon and should be ready to go soon.
Great, thanks so much. And we're getting a couple questions coming through. One question is, do you know what users are going to be accessing the portal and the data?
Well, we have been working closely with the Florida Keys National Marine Sanctuary and the Flower Garden Banks, and they are interested in specific views of the data. They're not interested in everything that's served through the portal. One of the problems that people that are in management positions have is, of course, time, and in many cases they're interested in just looking at change for specific variables, and they don't want to construct the analysis every time that you go through and enter the portal. So one of the things that we have been working with Axiom is that once you get to a point, you can save that state, and when you come back, you can reconstruct the analysis to where you were, or you can save the URL of what you see on the screen and then mail it to somebody, and whenever they plug that URL into a web browser, the analysis will be reconstructed for them. So it'll do the analysis, it'll show the time series, it'll show the right maps in the right area. So our understanding is that people in fisheries management are interested in some of the data. They are interested in fish distribution and how things move around relative to where there may be fishing pressure. The sanctuaries is interested in not only the environmental variables, whether things are warm or cold or whether they exceed a certain temperature threshold. They're interested in mapping disease and how disease may be spreading. If you have, right now we have a big problem in the Florida Keys where you have disease spreading and we don't really know where it comes from or how or why. So if you can map it over time, it helps understand if it's connected to a specific stressor, like maybe sediments or some other problem. And right now, so these are the kind of data that we need to get to put on maps and look at time series so that we can understand why things are changing. So if we have decline in coral reef cover in the Florida Keys, are there areas where that's more happening faster or slower? And if you have good maps where you can go back to and zoom in, zoom out, and overlay it with other data sets, is extremely powerful. But you cannot do that every single time with very complicated tools. So you want to have it in ways that are simplistic and simple to use, and yet tell a very powerful story. So I think that there's people looking at shipping, dredging, port development, tourism, people that look at harmful algae blooms in South Florida. They all want to know where it's coming from or where it's going. So if you can show it together with other data sets, including model outputs, that's extremely powerful. So I think that there's a large number of people that can use this.
Great, thanks so much, Frank. We have another question. Can you explain what seascapes mean again, and where is all of the data and results being hosted or archived for public access to use and also to provide feedback?
Yeah. Seascapes are a name that we gave to a thematic classification of a combination of different satellite data fields. For example, if we take a satellite time series of sea surface temperature and we combine it, overlay it, basically, we put one on top of the other for sea surface temperature, the amount of light coming from the sky, so what we call photosynthetically active radiation, and there's maps of that. If we combine that with maps of phytoplankton fluorescence, which can be measured from space, and these are, again, our maps and time series. And so if we overlay all of these, we can see that there are different patterns of chlorophyll concentration and fluorescence concentration when there's specific types of winds and temperature and depth profiles and so on. So we can, this is basically what is biogeographic regions, okay? But in the past, biogeographic regions have been pretty static. People try to collect biological data, they map it against depth and physical properties of the water, like temperature, salinity, and so on, and they come up with a construct saying, well, these groups of organisms live in this area, and we are going to call this biogeographic area number 1. But we know that these things change. We know that temperature, salinity, winds, currents, all of these things change in the ocean relative to topography and all of the in light. And so what we're trying to map is how these things change over time in a dynamic fashion. So that's what we call seascapes. So the idea is trying to understand what each color of a seascape means by validating it. So we've done that with, for example, the large marine ecosystem and looking at fisheries data using the sea around us fisheries databases, so that we can understand how fish catch and fish catch diversity maps against seascapes. So so fisheries data are funny in the sense that they're depending on. So the data depends on what people landed, and so it's not a fisheries independent number. But so that paper is actually published. The other thing that we want to do is we want to make these in an operational sense. So right now, Maria Cavanaugh at Oregon State makes these seascapes, and she's making them globally. So there's an effort to make these maps at a spatial resolution that is reasonable, like 9-kilometer resolution, and make them everywhere. And now what we're working with is we're working with a group called NESDIS, the satellite line at NOAA, and they have an office called Coast Watch. So Coast Watch has agreed to take in the algorithms. They collect all the data that we're using anyway, both from NASA, NOAA, and also European satellites. And so they're willing, and they're now going through the process of implementing Maria's algorithms using all these different data streams, and they're going to put it on Coast Watch, so it's going to be publicly available through the Coast Watch nodes. And so that's the way that we're going to release it to the public, and we hope that that's going to happen before the end of this year.
Great, thanks so much, Frank. We have another question. Are specific private sector partners envisioned for help in developing requirements, for example, the aquaculture industry?
Well, I hope so. I think that the aquaculture industry is already engaged in some of the regional associations on the West Coast, both for ocean acidification and harmful algae blooms. So I think that we need to understand exactly what products they may need that can be served through SECOORA, and I'm not totally sure exactly what SECOORA has done at this stage to reach this type of private sector. I know SECOORA has worked a lot with ROFS. ROFS is a fishing forecasting service that Mitch Roffer used to run. Now it's run by Matt Upton, so they use some of the SECOORA products. I don't know how much they are engaged in defining SECOORA products, but I can think that there are many opportunities to work with the private sector in developing sensors, but also in collecting and putting out specific products. But I'm not part of the SECOORA process doing that, so I cannot tell you. And hopefully, maybe somebody on the call is and can give a better answer than what I'm trying to do here.
Hey, Frank, I will confirm we do engage with the private sector. And actually, Mitch Roffer is asking, do you know or does anyone know what IOOS is doing to place biological sensors on NOAA buoys?
Well, there's quite a bit being done. NOAA, IOOS sponsored an ecosystem mooring workshop. My concern is that very quickly people are inhibited by the cost of the technology, and then they end up with a fluorometer on a buoy, which to me is not biology. And so we need to move away from the thinking that if we put a fluorometer on anything, that all of a sudden we're measuring biology. And so there's a mistake. I think it's an important and useful measure, but it's not a measure of biodiversity or something that can tell you how organisms in the ocean are changing. And that's what we need to understand. So there are these technologies that I just showed that NOAA is thinking about how can we get to a point where we can either drive the cost down or implement them in some specific limited areas so that you can get some information on how life is changing. So I know that IOOS is actively engaged in this. It's always a matter of money, and that's really where we have to come to grips with this. A lot of talk about the blue economy and developing sensors as part of that, and yet these sensors, the 2 or 3 that I showed, each one is over $100,000. So how can we make small microscopes that we can put in the ocean, or how can we deploy acoustic sensors in the ocean that can give us a better coverage? And that's something that I think we can do, but it has to be a community effort.
Great. So thanks so much, Frank. That was a great answer. And then we have another question. How could you include decommissioned oil infrastructure, also known as oil rigs, as observing platforms to collect data or to collect time series data of oceanographic information?
Well, I'm not involved in those discussions, but I know that there are people, the federal agency called BOEM, that is very much interested in advancing those discussions. So I would encourage those of you that are interested in that to contact Rebecca Green at BOEM for the Gulf of Mexico, or the regional science director for BOEM in your area. I mean, working with the private industry there is, of course, useful. And some people, maybe some people in the audience have the right private industry contacts. For example, people with Texas A&M are working with Shell and others to put sensors on not just the commissioned platforms, but also active platforms. So that's a big area of effort. A lot of people are involved in trying to use these platforms.
Great, thanks, Frank. We have another question. Can you provide more information on how MBON and ATN members are working together? For example, in the SECOORA region, the FAC network is collecting acoustic telemetry data. Is that being evaluated alongside satellite remote sensing data?
No, that's where we want to go, and that's why I brought it up, because I think that there's lots of opportunity to work together. So ATN is now developing its data portal or data service platform that's also being done by Axiom. It's the same company that does the data management and communication for SECOORA. So there are natural linkages. The same company has the 2 servers. The question is, how do you link those? What kind of gates do you put between the 2 portals so that you can talk to each other and get access to environmental data, or to get the animal tracking network data if you want to look at it in the combination of other biological data and environmental data. So that's where we want to go. ATN has been organizing workshops around the country. I think they've had 5 already, and they're going to have one in, I believe, in October or November on the West Coast, where they're bringing the local MBON partners to work with the animal tracking community in a workshop. And so if you're interested, you should look it up. The MBON ATN workshop is going to happen soon, and I think there may be other workshops happening after. This is a really exciting area of development, and I think we can all chime in and tell Bill Woodward, for example, and Gabriel Canonico how to better integrate those two efforts and what products we may want to see.
Great, thanks, Frank. And then we have one last question. Do you have a requirement for or interest in receiving credit for use of the data in publications?
Well, I personally put a lot of data out, and hopefully people will call me and invite me to be a co-author as they use it, and I always appreciate that and very happy when people do that. But, you know, we have served satellite data and time series data for a long time that a lot of people use in their publications. So I think it has, for me, it has opened an enormous amount of doors, and I have been included in many more publications than if I had not opened data up to the community. So I think that that's my experience. I, of course, would like to be invited every time somebody uses anything that I do, but if I don't, then, you know, that's the way it is, and it happens too. But I'm just giving you my perspective, and I know there's many different perspectives out there, but it does open opportunities for people to work with those that share their data.
Great, thanks so much. All right, we have time for one more question. Does anybody have a question they would like to ask Frank? All right, I am not seeing any, so Frank, I am going to take over controls and promote myself to presenter. And I want to thank you so much for giving such a great presentation. And I want to remind everybody that a recording of the webinar and along with a PDF of the slides will be at SECOORA.org/webinar-series. If you have any questions, please email communications@SECOORA.org or Frank. I put his email address in the chat box. And I just want to thank you guys so much for joining us. And Frank and Enrique, thank you for spending your lunch hour and giving us a little bit taste of what MBON is in our region.
Well, thank you, Abby. I really appreciate it, and thank you, everybody, for listening.
Thank you, Abby.
Great. Thanks so much. Have a great day.
You too.
Bye-bye.
Bye-bye.